So our objectives today are to get a feeling for why we use genetic testing. I want to do a little bit of, as I said, just kind of background on genetics, so we're all sort of on the same level. Because I don't know about you, but I haven't had a genetics class in over 25 years, and a lot has changed. I also want to go over some of the advantages and disadvantages of the different genetic testing modalities. And then we'll also talk about sort of specifically autoinflammatory genetics. And I'm tag teaming this talk with Ivona. So I'll be starting with a little bit of basics of genetics and a little bit about the genetics techniques and some of the pitfalls of genetic testing. And then she'll get into some of the other specifics of autoinflammatory disease. So why do we order genetic tests? Well, I think it's obvious. We want our patients to have a diagnosis. Ideally, we would be able to use that genetic test to give us some idea of what to expect for the patient. Genetics are obviously helpful for genetic counseling in terms of family planning and what to expect for future generations. And then I think one of the pie in the sky issues is that we could potentially, and this is again increasingly happening, we could potentially use genetics and then take that mutation and then decide how we want to treat them. However, there's a lot of pitfalls with this. First of all, a lot of times genetics is super nice, but may not really be that necessary. I mean, a lot of times we can still make the diagnosis clinically. In addition, a lot of times we get all this information back and it's really inconclusive. So oftentimes you're more confused after you get the test than you were beforehand. For prognosis, I mean, again, there are some situations where there's good genotype, phenotype, and you can make predictions. But in a lot of cases, we really don't know. That mutation doesn't really tell us what's going to happen because there's so many other things that are going on besides at the DNA level. A lot of patients are kind of too scared to know about genetics. And then the other question is, what do you do about incidental findings and other things? And it may not be that important for the family. They may not have any future children planned. And finally, I think for pharmacogenetics, we want to be there, and I think we are getting there, particularly in some of the oncology field, but we're really not there yet for able to sort of do that much precision medicine. So we'll start with a couple quick cases, and then Ivona will return to them at the end if there's time. But this is a kid that a lot of us see in routine practice, because fever is pretty much the most common presentation for pediatricians, and actually pretty common for internal medicine docs as well. But this kid comes with a fairly classic story of recurrent episodes of fever that happen very regularly every month and last about 4 days. Pretty much each fever episode is pretty much the same, starting with a sore throat, swollen glands, and just sort of feeling bad. Kid usually doesn't eat that well, doesn't have any signs of infection. So we don't see any obvious upper respiratory infection symptoms or any gastrointestinal signs. And the kid basically gets treated with Tylenol and Motrin. It helps a little bit, but then within the 4 to 6 hours of the medicine going away, you got to give it back again. And this kid is a kid who hasn't been exposed to sickness, and then other kids in the family are not getting sick from this kid. So, classic recurrent fever kid. Does this kid need sequencing? We'll come back and see what happens at the end. Second kid is a kid with an adult with cold urticaria who just developed urticaria at 21 years of age, has been seen by several doctors and given antihistamines for hives, and they're not working, but also has a lot of other systemic features like fever and joint pain and conjunctivitis. But this patient has no family history at all. So what's going on here? What do we do? And is this a patient where genetic testing would be helpful? And if it is done, will it end up being helpful? We'll come back to them later. All right, I want to start with sort of molecular biology 101. And this is a list of terms. And I'm not going to go through each of these terms, but I want you to kind of put in your mind a little bit, look at all these different terms and make sure you have a general idea of what's going on. I'm going to go through all these different things over the next 5 to 10 minutes. If there's questions, I'm certainly happy to answer them either during or after. But the idea is we're going to start from DNA and go all the way to protein. And these are all sort of the basic terms that are helpful when we're looking at genetic testing. All right. So we start with DNA. We all know DNA is made up of 4 base pairs, ATGC. We know there's 2 complementary strands. We know that DNA then gets associated with histones and get wrapped around, and that's what forms actually the form of the chromosomes. We also know these chromosomes in general exist in pairs, and there are short and long arms. That's sort of basic DNA and chromosomes. That's where we are with that. When it comes to what's on the DNA, that's the actual code, it forms these genes, and they are made up of exons, and then there are also a lot of untranslated regions, which are in the beginning and the end. Also, there's also introns in the middle, which are also untranslated. And then each gene has a transcriptional start site and a stop site, which is where you sort of decide where the mRNA is made. When it comes to transcription, that's when DNA gets transcribed into RNA. And in that process, there are a lot of things that regulate that, including sequences in the promoter region, which is oftentimes upstream of the gene, but also can be in these intronic regions. And there's also a lot of other things that are associated with the DNA that control how that thing is transcribed. And that's controlled by these things called histones, and whether or not things are methylated or not. And that regulates the level, and sometimes potentially splice forms and things like that are happening. But there's also a lot more to it as well, other than at the gene level. There's also these things called microRNAs and also non-coding RNAs, which also can have effect on other genes and have effect on a lot of different processes going on in our body. So it's not just simple, DNA is the gene, and then gene tells you what's going on. There's a lot going on at each different phase. All right. RNA also gets spliced, and the idea is you have these introns, which are not translated, that are spliced out, and then what can happen, though, is you can actually splice alternatively. So you can actually have certain parts of the gene actually completely removed. So then you end up with different splice forms of the gene, and those things can have completely different function or be completely non-functional. So now we've gone from what we think is a gene in DNA to now we have completely different forms. And so you may have a mutation in a particular region, but it may not matter because it may mostly get spliced out depending on the situation. All right. After RNA, we go to protein. And RNA is translated the amino acids, and that's what makes up the protein. We always have a translational start site, which is methionine. We have a stop codon. And then proteins can then be modified in a number of different ways, which can activate, deactivate, or change its stability. And so even though you have the gene, you have the RNA, and you may have your protein made, but it can completely change depending on all these different processes that are happening that are really not decided at the genetic, at the DNA level. So the bottom line of this sort of from DNA to protein is DNA isn't the only determinant. So at this point, a lot of what we have with this DNA testing or genetic testing is just what the sequence is. But RNA is expression is regulated in a lot of different ways because of all these different epigenetics and promoters and stability. You also can be very determined by which cells and tissue, and again, as I mentioned, the alternative splicing. And then once you get to the protein level, you also have a lot of things that are going on that aren't at the DNA level. The protein can be expressed differently based on its stability, how long it sticks around, or whether or not it's degraded. And then there are a number of different modifications that you'll learn about through more of this meeting that actually goes on at the protein level that isn't necessarily genetically regulated. All right, human genetics 101. We all remember back to our different inheritance pattern. We're just going to go through the autosomal and sex-linked, and we're going to go through recessive and dominant a little bit. And then we're going to talk about how we determine or define DNA variants, and that being mutation versus a SNP, the location of mutation, and then other genetic changes which are not quite as simple. All right. The issues is it's not always clear, and so we sort of have this idea of autosomal or sex-linked, but it's not always clear if it's recessive or dominant. And actually, we've been living for years thinking FMF is a recessive disease. We're rethinking that now, and so it really things really change around. And you can sometimes in one disease and in one gene have multiple different types of presentations based on whether or not it's recessive or dominant. And so it's complicated, and that's kind of the main point I want to get to. In terms of DNA variants, it's not always clear what's a mutation and what's a SNP. We also have these incomplete penetrant variants, which we'll talk about a little bit later. And then there's also this concept of somatic mosaicism, in which we have mutations that occur later on in development or even in adulthood that can totally lead to disease that aren't there present. So, so if you're not looking at the right tissue or cells, you may actually miss mutations in the patient. All right, a couple things about inheritance patterns. For autosomal versus sex-linked, if you have males affected only, it's a pretty good idea you have something sex-linked. If you have male-to-male transmission, then you're looking most likely at an autosomal dominant disease. If you're looking at recessive disease, you're likely to have skipping of generations. You also commonly see in certain populations where there's a lot of consanguinity, you see more recessives, whereas dominants, you don't see that. There's also the concept of this gain or loss of function. And that oftentimes is it can be a debate, because you can sort of have a loss of a inhibitory function, which can look like a gain of function. And there's a whole concept of dominant negative, in which you have a mutation that actually is a gain of function, but is actually leading to a negative effect, then causing an inhibition. So these sort of things get tossed around a lot with gain or loss and dominant negative, but you can sort of think about it a lot of different ways. So autosomal and sex-linked are not always clear. Same thing with recessive and dominant. And again, you can have this incomplete penetrance, which can mess up the whole sort of concept. And again, all these different ideas about gain or loss of function are not completely clear. All right, here's our basic pedigrees. You remember the autosomal dominant, that's up in the upper left corner. And there you're going to see no skipping of generations. You're going to see male-to-male transmission. For autosomal recessive, you're going to have parents who are potentially unaffected, so those are your carriers, and then they have either affected kids, carriers, or unaffected kids. For the X-linked, you're going to see mostly females as the transmitters and males as the patients that are affected. And then in very rare situations, you have the X-linked dominant. I'm not even going to go into that because it's so rare. All right, but here's sort of a general idea of how things sort of happen. When you have a dominant disease, you have a 50% chance passing it on, and if you have an autosomal recessive disease and two carriers, you then have a 25% chance of it going on. It's all because we all have two different chromosomes that then sort of give you the options either way. All right, with the X-linked, you have the carrier mother, and with the X-linked, you have an affected father who then can pass it on to daughters, which is extremely unusual. All right. What we talked about mostly in terms of these inherited things are situations where mom or dad passes it on. The other possibility is de novo mutations, and we see that a lot in autosomal dominant diseases. But the other thing we see primarily in these autosomal dominant diseases is this concept of somatic mosaicism. And what happens is there can be a somatic mutation that occurs at different points of development. It could occur very early in development, in which case you would have a large number or percentage of your cells with that mutation, but can also happen later on in life, and even just particularly in one particular cell line. So it can be just in myeloid cells, like we see in some of our patients with autoinflammatory diseases. And in some situations, you may have as small as 5 or 10% of a particular cell type, like a myeloid cell population that's affected with mutation, and that can actually give you a phenotype. It may be a later onset phenotype, it may be a slightly different phenotype, but can also be a full phenotype. This causes all kinds of problems when you're thinking about genetic testing, because one, you may be getting a blood test or a saliva test to get your genetic test, but you may not be looking at the right cells, or the number of cells that are actually affected may be so low that your genetic technique may actually not be able to detect at a level of sensitivity that can actually pick up the mutation. It also completely messes up our ability to do genetic counseling, because in a lot of ways, we would often say, okay, we think your child has an autosomal dominant disease that is that obviously is a de novo mutation, but we may be wrong, because it could be that the parent may actually have a gonadal mosaic, and could actually have another child with the disease. And so tons of different situations where it sort of messes up our whole idea. All right, now to mutations. Usually in my talks I show surfing pictures. This time I'm only going to show you surfing as a word. Anyway, but what happens in different types of mutations is we have substitutions where you have a single change in a base pair, and you would change then your spelling of surfing. You can also have insertions in which you have an addition of a base pair, or could also be 3 different base pairs, which then wouldn't lead to a problem with translation. You can have a deletion, and that can be a situation where you just lose 1 or something in divisibles of 3 in which you just lose don't mess up the frame of the translation. Or you can have a complete truncation where you have an early stop. And those are sort of the different types of mutations that we get. Problems, well, you can have a silent mutation that does absolutely nothing. It could even change the amino acid but still not actually have any major effect. Or you could have a change in the frame, but sometimes that can be corrected as well. All right, where are most mutations? Most are in the exons. There are some that are in the splice sites, and they may not affect the specific translation, but could actually affect how many exons are actually put into this alternative splice form. And then again, there are also mutations that can occur in promoters or intronic regulatory sequence. And that makes a difference depending on the type of genetic sequencing technology you're going to use. All right, so what are some other genetic changes? Well, you could have deletions. You could have just a few exons removed, and we've found that in some of the diseases that we'll hear about later. You could also have copy number variants in which you have entire genes that have been duplicated or entire genes that are just deleted. Or you can have really large deletions and duplications or entire chromosome changes as well. Different techniques, different genetic technologies are better or worse at sort of picking up these different types of genetic changes. All right, so how do we do genetic testing? Old school, we used to do very mutation specific, and that was either done by hybridization or done by allele-specific PCR. That's not used very much anymore. The old school way of sequencing, which still is considered sort of the gold standard, oftentimes is used to sort of confirm something, is Sanger sequencing, and that's usually an exon by exon type approach. But what's happening now, in pretty much all of the many of the research and now commercial testing, are these next generation sequencing platforms, and this is really the way we do things, and that can either be used in terms of gene panels, which we'll talk about, exome sequencing, which we'll talk about, genome sequencing, or even transcriptome, where we're just looking at the RNA as well. And so if you look at sort of all the different clinical testing sites, there's a number of out there. Most of those are now using next-gen sequencing. But there are issues with these doing these clinical genetic testing in terms of getting insurance to cover it, as we all know, and that's a big issue we're hoping to solve in the next couple of days in our working groups. And then also, when you do genetic testing, are you prepared to deal with that in terms of genetic counseling? Because you may end up with learning a lot of things that you weren't necessarily expecting or not know what to do with. All right, PCR is sort of the basis of a lot of this stuff. And is sort of the basis of being able to reproduce DNA. It's used by creating these primers that surround the area you want to amplify, and then you can then either see that amount of DNA or you can actually sequence it, and that makes a big difference. Again, Sanger sequencing is based on sort of PCR. It's an idea where dyes are put at the end of each different oligomer as it's sort of created, and that's how we get these little peaks at the bottom down there of different colors. And that's how we base by base can sort of look at something anywhere from a few hundred base pairs long to maybe up to 5 or 500, even 1,000 occasionally. But we're sort of limited in our length of what we can do there. With NextGen, it's actually much more a shotgun approach. You actually do a number of different amplifications, and then you just tile them all together and hope to get enough coverage so you can actually trust your sequence. And there's a number of different ways to do that, 454, deep sequencing, things like that. But it's all based on sort of getting genome DNA, capturing it in some way, and using PCR in order to give us a lot of different things. But there's actually a newer technology, these long read technologies, which give you the ability to get really long pieces of DNA. And that can reduce the error rate, allow for easier assembly and better accuracy. and also help you with copy number, which is a problem that we sometimes see, and can also allow for transcriptome sequencing, and that may be something we see more, which then allows you to figure out these splice variants and things like that. It may also, because it's big enough, be able to let you capture haplotypes in terms of different DNA, large stretches of DNA that actually seem to run together in different populations. All right, the problem with all this exciting new technology is that we are getting tons of information, but we don't know how to deal with it. And the real problem has been that the cost has come down so quickly, faster than we're actually able to compute it and sort of deal with it in that way, and faster for our minds to be able to sort of deal with it. So this process of generating data can happen really quickly. At our institution, they can generate a sequence within a day and hope to have it analyzed within a couple days. But the real problem is that sort of human part at the end, where it requires sort of a human to sort of do it. And what's happening more and more is AI and people are actually getting the computers to do more and more of these last steps of the process. But still, you need someone to think about it. And that may, again, come down to computers in the future, but that's where we're at. So the main bottleneck is really this data analysis, and so we're still trying to sort of figure out how to deal with what to deal with all this information, either if it's positive or negative, and we really are drowned a lot of times in this next generation. All right, in the last few minutes, go through just sort of what we think about whole genome versus exome versus gene panels. So on the top of this slide, it sort of gives you, those of you who are readers and think about, I just want to give you an idea of the magnitude of this stuff. For those of you who've read War and Peace, I haven't actually, it's 3 million letters. But if you're going to do an entire genome, a whole genome sequence, you're looking at 3 billion bases, so that's 1,000 copies of War and Peace. So to bring your DNA sequence, your whole genome sequence to your doctor, that would take a truck to sort of bring it in. All right, exome sequence is a little more manageable, but we're still talking about 10 books of War and Peace just to sort of get through the whole thing. Now the gene panels are a little better. That may just be a few chapters of War and Peace, but still a lot of DNA and a lot to sort of go through. Now if you're not a reader but you're a sports person, this is kind of how it works this way. For a genome, you basically watch the entire football game from the beginning to the end. You get every little detail, every little play that's happened, all that kind of stuff. And exome is like reading about the next day. You sort of hear about all the highlights. It goes through what happened in the first quarter and the second quarter. But if you're doing these panels, then all you're really doing is just focusing on either one or two players and sort of seeing what they do. So you get a sense for the magnitude of sort of what you get with these different techniques. All right, I won't go through this because it's a lot to sort of go through, but the concept is that with Sanger sequencing, you're limited to really a few genes. You're limited in your size, and the cost is relatively cheap per each sequence, but again, you're not getting the kind of information you get with these panels, in which you can get sometimes up to 300 genes in a panel, and you're able to get a fair amount of depth for these things as well. For whole exome, you're looking at a total of 20,000 genes. Most of the coding regions are covered pretty well, but not always. There's always some areas that aren't really covered that well. You can get fair depth, but sometimes you're not that great. And when you get to whole genome sequencing, you are dealing with not just the 20,000 genes, but all the other stuff as well. And again, you don't get quite as good coverage a lot of times, and costs are coming down to the point where whole genome is actually less than whole exome in a lot of situations. All right, so this is where I pass on the baton to Ivona, and she'll talk a little bit about how we take all this information and deal with it in autoinflammatory diseases. Thanks so much.